We're going to talk about Introduction to Y-DNA today. And in case you aren't aware, my name's Kelly Berkheimer, and I have a virtual genetic genealogy group, DNA group, that's called Genetics, Genealogy, and You. You can find information about us on Facebook. You can join the email list and find out when the next meeting is. Currently, we're meeting the third Sunday evening from 5 to 7 p.m. Eastern Time Zone. So we'd love to have you join us sometime. It's completely free. The group is free, the recordings are free, and the email list is free. So we're going to talk today about Introduction to Y-DNA. Y-DNA is one of those areas where I do not feel like I am my strongest. Most of my strength is in autosomal and mitochondrial DNA analysis and using that to help adoptees find unknown parent or parents, and also in using it for genealogical purposes for other finds and other brick walls in our tree. But Y-DNA testing is something that I consider to be a pretty big specialty. Each surname group or project kind of runs differently. We'll talk about those later in the talk, but my only experience with Y-DNA has been with my husband's Y-DNA kit. And so when you have very limited experience, I like to at least tell people that I don't consider myself to be an expert in this, but I have learned a few things along the way, and hopefully you'll find something today that's really helpful for you. So we're going to talk about, we always start our DNA talks with the cautionary tale. You might find surprises. Anytime you do DNA testing, you might find out things that you didn't know. You might be the secret keeper that ends up finding out something and then trying to figure out what to do with that knowledge. Also, you might find, particularly in Y testing, that your surname is not your surname. It's quite possible that you will take a Y-DNA test and you will find that your match lists have surnames that are different than your own. Maybe there are some with your name and some with different names. So that's a pretty common occurrence. Don't let that freak you out, but it is a possibility. Also, you may think that you don't have any matches at all. I hear that every once in a while from people because they don't have any close matches. And the DNA testing company, FTDNA, Family Tree DNA, will only show your matches out to a certain amount of genetic distance, and we'll talk about that. But that doesn't mean that you don't have matches. You do have matches. They're just not close enough to be genealogically or historically significant. But remember, all men are descendant from one man. So you do have matches. You match every man who has lived. You might also not have any genealogically relevant or current time frame matches. That does happen sometimes. There are a lot of reasons for that. It has to do with who's tested, how many men are in the database. It can also have to do with when your person or that line immigrated to the US and they're testing along with these other US men, or predominantly US men. So if you've taken a Y-DNA test and you don't have very many close matches at all, it may be because you're working with a recent immigrant line. That's the case with my husband. He didn't have very many close matches at all. You might need to wait for more close matches. We do this all the time with our DNA analysis. It's really important to be organized, keep good records, so that when a new match comes in, you can pick up where you left off. And don't forget, just because I've given you all the discouraging things that could happen, there is a really encouraging thing that could happen. You might find key answers. You might stumble upon a group of men who are researching all the different lines of your surname in the US, or you might find a wealth of database information and match information. So you never know what your results are going to be, and you might need to wait, and then all of a sudden there are some key answers. We're going to talk today about chromosomes, but specifically the Y chromosome. So if you look in the lower right corner, the Y chromosome is one of the the sex chromosomes. So if you're male, you are X and Y. If you are female, you are X and X. And again, we're not talking about gender in this particular case. When it comes to DNA, there really isn't such thing as gender. There is sex. You're either male or female based on whether you're XY or XX. The other 22 pairs are what we call autosomes, and that's where the word autosomal testing comes from. And in autosomal testing, it does get a little bit confusing because companies do test the X. So that's kind of thrown in there because both men and women have an X. Women have two, men have one, so they test that also. But we're going to talk today about the Y chromosome. But before we move on, I do want to point out that each one of your cells has 23 pair of chromosomes, but they're not all pretty and lined up and paired up like this. This is what's called a karyotype, and scientists will drop a cell onto a microscope, take photographs, cut out the ones, the twos, the threes, pair them up. And so that was a pretty old school thing we were doing in the 1980s when I worked in a genetics lab. And so this is not the way the cells appear, or the chromosomes appear in the cell. So just for your information. There are three kinds of genealogical DNA tests, and those can be best visualized using the pedigree chart. So if you look on the left where there's me, let's start with the Y-DNA test. Now, I cannot take a Y-DNA test because I'm female. I don't have any Y-DNA. So my dad does. So I could have my father test. I could have my brother of the same father test. I could have my father's brother test. So any of the males in that line who are descendant from the same fathers carry the same Y DNA. So it's the top line of your pedigree chart, my dad and my dad's dad, and so on back in time. And moving forward, all of their descendants moving forward of those particular men. There cannot be a woman in that line of Y DNA testing. It has to be male, 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 male. So sometimes we call that the surname line, or the top line of the pedigree chart. Mitochondrial DNA test, at the bottom you can see, would start with me because whether I'm male or female, I do have mitochondria that I received from my mother, and she received from her mother, and she received from her mother, and so on. So there's an unbroken female line, but remember at the end of the chart on the left, it could be a male or female who received mitochondrial DNA from their mother. Every person has mitochondrial DNA, but only women pass it to their children. The autosomal DNA test is a small piece of each of the people who make us who we are. And so when we're talking about maybe solving a brick wall problem and which tests to take, we want to think about it in terms of this chart. If it's mom's line, we might want to consider mitochondrial testing or autosomal. If it's dad's dad, we might want to consider Y testing or autosomal there too. But if it's any of the folks in the middle, who are all equally valuable in making us who we are, we want to consider autosomal DNA testing. So remember which tests, which DNA. Your Y DNA matches might have clues in the autosomal matching. So we're going to talk later when we look at Family Tree DNA's site. We're going to look for that FF, which is the name of their test, Family Finder, in the match list. So that would tell you which of your Y DNA matches have also done the autosomal test. And so you will have matches who match you on both. And we'll talk about the cases where you'll have matches who match you on one and not the other. So remember, you can have a valid Y DNA match who does not match your autosomal DNA. So we're going to go back to some of the things that you've learned about in your basic genetics and your autosomal testing. All first cousins and second cousins will share segments of autosomal DNA. There's never been a proven case of first or second cousins that share 0% of autosomal DNA. Now, third cousins and beyond can be biologically related to you, but not share DNA with you. So two third cousins can share 1 to 2% of DNA, but sometimes they share 0% of DNA. So there's a wide range of how your third cousins can match you, and that has to do with how the DNA is recombined. So recombination affects the pieces of DNA that we get further and further back. A third cousin would receive about 6% of their DNA from their second great-grandfather. So you've also have to consider that they've missed 94% of that man's DNA. So you might match a man, if you are a man, you might match a man, a second great-grandfather, with Y DNA testing, but you've only received 6% of his autosomal DNA, or less, or a little more. It varies. So you might have missed a pretty good significant amount that another cousin could have inherited. So it is possible for two men to have Y DNA that match and no autosomal DNA that matches. So we have to talk about phylogeny in order to talk about haplogroups and how the group of all men are related to all men. This also is a phenomenon in biology where certain animals are related to animals, plants are related to plants. So it's a common biological concept that a phylogenetic tree shows inferred or evolutionary relationships between DNA of a specific animal. And it's based in similarities or differences in DNA samples. It shows an inferred common ancestor of a certain genetic distance to one another. And each animal that's at the terminal end, so at the far right, has similarities, but it has a difference from the group that preceded it from the left. So let me just explain. This is a quick, not complete, phylogenetic tree for dogs. And so if you think about over time how dogs have changed, maybe become from wild to more domesticated, there are wolves and coyotes and wild dogs and all kinds of regional differences based on climate. And so there are adaptations that have happened where we have lots and lots of different varieties of dogs. This is the same thing that happens with human males in a in their Y DNA with a haplotree, and it's the same thing that happens with all humans in the mitochondrial haplotree. So let's talk just for a minute about the inheritance pattern of Y DNA testing and what that test discovers and what that test will tell you. So only men have Y DNA. It tests the male line, sometimes called the surname line. The Y chromosome is passed along intact generation after generation after generation, sometimes unchanged for tens of thousands of years, and there are about 58 million base pairs. Now, there are changes that occur, and those are called mutations, and along the way, enough change has happened that a new haplogroup is discovered or set aside, and so the tree ends up branching and branching and branching. So let's look at just a rudimentary, simplified look of the Y chromosome haplogroups. If you've ever gone to Y-DNA to your site on FTDNA to look at your own specific haplotree, if you haven't done that, I recommend you do it. I have some screenshots in a little bit of what that looks like. But of course, every man is different depending on which haplogroup they're in. So a haplogroup is a genetic population of people who share a common ancestor, and there is a patrilineal and a matrilineal line. This is showing the Y DNA, which is the patrilineal. Mitochondrial DNA would show you the matrilineal. So the Y DNA haplotree starts on the left with the group A, and there are other subgroups under A. and it goes all the way to the right, which is more European, UK. The oldest haplogroup, A00, is the oldest, and that's Y-DNA Adam. And we don't talk about Adam necessarily in the biblical sense, but we talk about Adam in the first man sense. And so as you move across from A to R's, what you'll find is that this very nicely mimics the general migration pattern. And we'll find out later when we talk about my husband's specific results. Right in the middle here, the letter I is where my husband's group is. And it's a common group. He's a Bergheimer. It's a common group for German, but it's also a common group for some others. And I'll show you that on the migration map. So depending on where your particular branch of the tree branched off, these are going to be the major headings of haplogroups, and there are much more detailed haplogroups below. So this is just a map from Family Tree DNA that shows the basic human migration. So if you think about where was the A group, Adam, you can see in Africa, sort of the left center of the screen, it says Adam, and that's where the locations have been for the presumptive Adam, the original Y-DNA. And so you can see them, how there's different migration patterns. Sometimes there's a northern branch that goes up into Europe. There's a branch that goes, the Q branch is fascinating. That one goes all the way across Russia, Siberia, down through Alaska, and down into Central and South America. Fascinating migration group there. And as you can imagine, there's a lot of indigenous who are in a Q haplogroup. And they were some of the first, not only Native Americans in the US, but indigenous Americans in the Americas. And so there's some fascinating histories about this. You can always Google haplogroups. It's fascinating, particularly Google your own, but then look at some of the other migration patterns and where did people come from and how did they end up where they are. There are some places you can investigate the Y-DNA haplogroup phylogenetic tree. Y-DNA haplogroup tree is a place that I look quite often. Family Tree DNA has a lot of information about the phylogenetic trees. And so let me show you a couple of things in a few of the future slides that you'll want to know. We need to talk about genealogical relevance for just a minute because, you know, we have a lot tied to surnames, and surnames are a fairly recent phenomena in history, not necessarily old history. So when we talk about Y-DNA results, we're talking about deep past, which is where those haplogroups were formed, migration patterns started, and then we have the time period that we call historical lineages. And during that time is more where the surnames might come into play. And then we have what we consider to be recent genealogy. So recent genealogy might be the first 5 or 6 or 7 generations on our tree. But pretty soon we find ourselves into historical lineages or biogeographical information where we're tying back to a region, not necessarily to a specific man or a specific couple. So there's a point where the surname line tracing and the Y-DNA tracing kind of puts us in a larger group, but not necessarily with a particular man. This blog from Roberta Estes, DNA-explain.com, has a couple of great graphics for you to look at. I included it here because you can go back and read about the case study that she did on the Kenny line. And I love this visual because it does show over time how a mutation can happen. And then when you share common ancestry and the Y DNA is passed on, it can go hundreds of years with no mutations. And then there might be a mutation, and then there's hundreds of years again without a mutation, or maybe it happens one generation after another. So you can read more about her blog here, but she's talking about people who all link back to the same man, John Patrick Kenny. And over time, there are mutations. So if you see the first generation there, there's a Paul Edward Kenny who has one mutation different than a brother. And moving forward in time, hundreds of years sometimes, there are mutations that occur. So when John McKenney had two sons, one had a mutation, one did not. And so you can see how four distinct lines develop over time. Now, those all link back to the same John Patrick Kenny. They're just four different variations on that Y DNA. And sometimes this is hundreds of years that have passed between generations. We're going to talk a little bit more about mutations in a little bit. We do want to talk about, in terms of this article, one thing that Roberta Estes says is that in the span of time between 1478 and 2004, this group of Kenny families had four distinct lines, and there was one line with no mutations at all that was exactly the same as the furthest back ancestor that was recognized. And some had one mutation each, some had two mutations, and these mutations are called line marker mutations, and they allow the testers, or particularly the project managers, to quickly and easily see which line of the Kenny family someone might descend from. So you can see how this might help you in a brick wall situation. So I'm just pointing out, you might want to go to see and read that blog. It's a really informative look at a long-term Y-DNA group of men. This is a picture of what I use to teach about genetic distance. And I just made this chart myself. So this isn't based on a real case, but if you talk about you as the man in the lower right corner, and you've got your dad and your grandfather and your second grade, third grade, fourth grade, fifth grade, you can see that going up that line, all of you, by the noting green, all have the same Y DNA. There's no mutations, there's no changes, and so all of you are an exact copy of each other. If you look at the next column over from you, going left, your fourth cousin who tested, and his father, and so far back that ties back to the fifth great-grandfather, are all still genetically identical to you. That's a genetic distance of 0. So everyone in green is a distance of 0 to each other. So that's why when you see your Y-DNA results, if you happen to have any genetic distance of 0, it's difficult to know. Are they close in time to you? Are you all connected further back? When did the mutations happen? It could have been very far back. So that's what makes the Y-DNA analysis so challenging. Now, if you look at the ones in light gray, you can see that the great-great-great-grandfather, so the third great-grandfather, had 3 sons. 2 had Y-DNA exact copies of him, but 1 had a mutation difference. And so I've colored those gray. And then that mutation is passed on to son and son and son and son. So you and your fourth cousin are 1 genetic distance from one another. You have a 1 mutation difference between you and your fourth cousin on this line, but you are a 0 genetic distance, perfect match to your fourth cousin who is in green on the other line. So it is possible to have 2 fourth cousins you match, and one is a genetic distance of 0 and the other is a genetic distance of 1. And then you can see, if there's mutations further back, how you have other cousins who come out with 2 genetic mutations or 3 genetic mutations, and so on. So I have a couple little summary things here. So we're going to talk about STRs in a minute, but the way that the testing companies place you into haplogroups is with what's called an STR, a short tandem repeat. And I'm going to show you some pictures of those, and so you can kind of wrap your head around that. But the short tandem repeat is a set of bases that repeats over time. So it might be an ATT, ATT, ATT. It doesn't really matter that much what the bases are, but the fact that they've repeated 11 times is significant. And then perhaps moving forward, there is a mutation, and so that repeat happens 12 times instead of 11 times. And then eventually it might happen 13 times. So those are genetic distances that can be captured by your test results. So remember, if you are in green here, the genetic distance of 0 from you are all the other men who are in green. The genetic distance of 1 from you is anyone who is in the gray category, but also any of the cousins in the yellow category, because they only are 1 genetic distance away from you. There would be a case of 2 genetic distances if you look at your 4th cousin that's in gray and how that 4th cousin is related to the yellow. So the gray is a short tandem repeat of 13, and if you subtract 11, you have a genetic distance of 2 for how the 4th cousin in gray is related to the 4th cousin, or to the cousins in yellow. So genetic distance has to do with where the change happened, but also how close are you to the change, 1 or 2 or 3 or more away. So let's look at your results page at FTDNA, so you can kind of get your head wrapped around the fact that 750,000 men have tested, and here's what those pages look like. So I'm going to show you the FTDNA results page from my husband. That's the only test that I really have access to. And so you can see here on what I've circled is the paternal ancestry. That's the haplogroup to which he was assigned. I will mention that initially when he tested, he was in a different haplogroup, and I am 253, and we're going to talk about how that happens. But there is a haplogroup there of IY5497, and that is taking into account 2 pieces of information: the STR, which is that short tandem repeat we talked about, and another further test called a SNP test. And we'll talk about both of those so you can see what that looks like. But the testing, the 2 different types of testing, push you further and further out into the branches. So the STR testing will give you an overall haplogroup, where you placed, are you in the I's or the R's or the A's? And then the SNP testing will push you further out on the branch to figure out how far out does it go before you reach what's called a terminal SNP. So where the end of the branch is for now. Obviously, as more and more men are born and more men are tested, we find out there are more and more terminal SNPs. And so that changes over time. It's changing quite often. The upper right-hand corner, you can see that my husband did the Y37 test. So the Y12 and 25 are some of the older tests that are included in your results. 37 is the lowest test that the company offers right now. And my husband did the 37 test. There also are 67, 111, and Big Y beyond. So we'll talk in a little bit about the differences in those tests and why you may or may not want to move on into those higher resolution tests. So let's look at what you can see here from the results page. So look at those categories in the middle and read those so you can know the kinds of information available. You can see there are matches, advanced matches, haplotree and SNPs. That's the screenshot at the bottom is what the haplotree and SNPs looks like. Also ancestral origins, so you can learn some history about the haplogroup. Also haplogroup origins, fascinating. Look at the migration patterns over time, migration maps. There are matches that have maps. There's SNP maps, lots of different things. There are two things I do want to point out. Well, one in particular that we're going to look at towards the end. The print certificate click will take you to not only the STR certificate, but the SNP certificate. So you will want to print both of those so that you have those for your records and you can use that to analyze your matches. There is a data download button where you can download your match lists and similar things like that. So it's a lot of great information here. Don't bypass opening all of these things just because you're excited to look at your match list. These other areas will help you understand. At the bottom, what I did was just a brief screenshot of my husband's group on the tree. And you can see those vertical and horizontal lines on the left. Those are the major parts of the haplotree. And the IY5497 is where his branch is, his terminal SNP is at this moment. This is kind of what it looks like. I tried to, the entire thing would be so small you wouldn't be able to read it. But I tried to pick out kind of some critical pieces along the branch here. So this is, you can see that the IZ63 at the top is where I started. If you keep going, the IZ40, that's at the bottom of the first screenshot, is the project that he's placed in, but his group is further down the tree in the IY5497. So I've kind of broken up the chart a little bit so that you can see how deep it can go. It can go really far. There is a place in the middle on the report for variants, surnames, and countries. And what's shown here are the variants. So what are the different terminal SNPs that show up? That's what those letters and numbers mean. You can click on surnames and you can get names here instead of variants. And you can click on countries. And I think I have a screenshot later of what it looks like when they put all the country maps in there. When you click the three dots, you can also see, there's always three dots you can click, and anytime you're on a website and you see three dots, you should click and see what there is there. You can see the country report for his Y-DNA haplogroup. Not surprising to me, the largest number of people in that group are German, and there are 10 people in his branch, and then downstream, further SNP testing and others are, there's a 520 people further down. And roughly 37% of the people with this haplogroup are German. There are some in the United States, of which my husband is one, so he's one of 5 there in the United States. And you can see there's other countries represented. If you look at the migration map, you won't be too surprised that some of those countries appear. But obviously there's a lot of time that's lapsed and a lot of migration that's happened when those haplogroups were formed. This is a map that I generated on SNP Tracker. I've included the link, and the link is in your handout as well. But SNP Tracker is a website where you can put in the terminal SNP, and you can find out the general migration pattern. And again, this is a general migration. This is not the way that they all went. You know, there's nobody saying, hey, all of you who match my haplogroup, let's go this way. There's no one saying that, but starting with DNA atom and moving up into Europe is how that haplogroup moved. It's a pretty old haplogroup, the one that he has, that my husband has, Neolithic, pretty old. You can see here, the reason I'm showing you kind of this blow up of the map is when my husband first did his DNA testing, he was placed in the haplogroup IM253. And he was in that haplogroup for a long time. He was first tested in 2017. We did SNP testing shortly after, and they still had him placed in IM253 for quite a while. It's only recently that they've moved him out on the branch to IY5497. But if you look at where M253 comes into play, it's a pretty large haplogroup. I'm often talking about this at conferences or presentations, and people come up to me and they'll say, I'm in that group, or my husband's in that group, or my father's in that group. So when you think about migration, you can see here that the IM253 has ended up going north for a bit and then coming back down into what's considered modern day Germany. Now, of course, modern day Germany is not necessarily what old Germany was, but my husband and his Bergheimer line are German, at least as far as we know. This is what the report looks like if you look at the countries. And I thought this was fascinating because you can see the number of men and the countries they're from and who they've tested with. And I just think that's fascinating, all the different flags and everything. But I do want to point out that there is what's called a SNP path, and it'll tell you, at the point of branching, what time period, what relative time period that was. And they're using BCE and CE, which is the newer way to mark time. Most of us probably grew up at a time where it was BC and AD, and that had to do with before Christ, or Anno Domini, which is the year of our Lord. And so rather than have a religious reference to it, it's the exact same thing, it just takes out the religious reference. So BCE is before Common Era, and it's formerly known as BC. So if you look at where my husband's line is, the first time he did the testing, he was placed in the IM253, and that's about in the middle of the chart, about 3,000 years BC or BCE. And now he's all the way at the bottom. His terminal SNP is IY5497. So somewhere in the 2000 to 3000 BC range is when his haplogroup differentiated. And again, these are estimates, and they're just scientific, educated guesses. But his first marker test, and then after SNP testing, more men tested. He moved down into the tree, and now he's in a project for I1Z140. And if you look at the chart, 2 from the bottom is the IZ140. That is the surname group or haplogroup project that my husband's been assigned to. It's not really a surname group, it's a haplogroup. And so he is placed into that project because he is further down the line. So all of his genetic changes include everything from the bottom line here all the way back. So his Y-DNA matches actually all of these men to a certain extent. So this is the 253 where he initially tested. This is the IY5497. And then there's an IZ140 group that he is in now and just recently in. So I don't know a lot about that project yet, but I am investigating. So if you look at the results for Chris, what you'll find is that his closest genetic distance match is a 3. Now he took a 37 test, and this was at a point where they had his haplogroup at the IM253. Since then, some of these men have differentiated into other groups. But his closest match was a genetic distance of 3, and the other matches were genetic distance of 4. So you might look at that and be discouraged and say, you know what, I just don't think that's going to be all that relevant. But stay with it. Stay with it. You never know what you're going to find. I want to talk about some of the symbols that show up in the match list so that you know where you can find more information. There is, with each of your matches, a message icon. That's what that envelope is with the little piece of paper in it. So you can shoot messages to your matches. You can talk about, who's the most recent ancestor that you think you have in common, those kinds of things. There is a button that's called TIP, T-I-P. It's a report that shows the probability of matching within a certain number of generations. I'll show you that on the next slide. There's also a notepad, so you can keep notes about your matches, that maybe you contacted them, they have a common ancestor of whoever, you can make notes back and forth. And also, you can see here that both of these have FF for Family Finder. So Family Finder means that they've also taken an autosomal match test. So you're going to want to go to their autosomal list for your husband and check to see if he is an autosomal match to these matches. Now, chances are genetic distance of 4, you might not find any information. Always worth a look, though. And then you can see which test they took and then if they've done the Big Y project. So the Big Y project notes all of the Y DNA, and it's a pretty common test for those who are super into this. You might be asked to upgrade to that by a project, and it's a pretty expensive test, and most of the time it's not necessary to do that, but I just want to let you know that that is an option. Also, you might see the little purple icon for GEDCOMs. A lot of men on my husband's match list uploaded GEDCOMs, and so you can see what they believe is their family tree. And this is the TIP report. I just wanted to show you like a screenshot of that. So on the 37 marker test, it'll talk about the probability between a match and you, or a match and your husband, or whoever's done the test. So this shows that the match is roughly 33% likely to be within 4 generations. 73% likely to be within 8 generations. But how many of us have 8 generations on our tree, particularly on just that one surname line? Not very often do I see 8 or 12 generations that people have used genealogical proof standard to determine. So that's just something to consider, that these matches that are 3s or 4s could be very, very far back. And it doesn't mean that it's not relevant, because in aggregate, that tells us a lot. But individually, you might not be able to tie it back to a specific ancestor. So what's the difference between the 37, 67, and 111? Blaine Bettinger always uses the example of resolution. He uses an example of a TV and how, black and white versus color, and the different kinds of televisions. It will give you more specific and information than what you had before. And so I think of it as sort of looking out the window with and without my glasses. So I can look out and know that there is a cityscape, but I have to put my glasses on to see the buildings. So it's about resolution between those tests. So let's talk about the genetic distance and the number of mutations that are reported depending on which test you take. And that has to do with resolution. It has to do with the specificity of the test. So you noticed on my husband's Y37 test that he only had matches that went out to 4s. That's because that test does not have enough STRs represented to show other results that are further out. If he did a 67 test, then all the matches that are 0 to 7 genetic distance would show up in his list. If he did a 111 test, all the matches genetic distance from 0 to 10 would show up in that list. So what I recommend to people when they initially start testing, the 12 and the 25 are not available right now. Those are kind of legacy tests that are still there. So start with the 37. If FTDNA will keep your sample, and you can upgrade to 67, you can upgrade to 111, you can upgrade to Big Y. So there are lots of options there, and sometimes they'll pull one and they'll say, well, we're not going to offer this one anymore, only these. So you can always upgrade at any point. Once you've done Big Y, though, you're pretty much done with your Y testing. There's not a test beyond that. You also might be asked to do SNP testing. So This, we'll talk about that in just a minute, but this 12, 25, 37, 67, 111 can get confusing because on a 37 test, someone could match you at 37 but not at 12, and that has to do with genetic distance. So if your match is a genetic distance of 3 and you have a 12 test, you wouldn't see it because they're only showing you the zeros and ones on a 12 test. So you do have a match. You do have a match of someone, genetic distance of 3. It's just that the company is not noting that in your match list. But you would see that on a 37 test because if you match a 12 test at 3, you're going to see the zeros, ones, twos, threes, and fours. So that just has to do with the testing algorithm and the resolution and specificity of the test and how they display the matches. I recommend you start at 37. Then what the company will tell you is if you do need further SNP testing for them to place you further down in the list or further out on the branches, then they'll tell you you need more SNPs. So let's talk again about STRs and SNPs, and let's do a little visual with that so you know what I'm talking about. We're actually going to show this slide again in a little bit, but short tandem repeats, STRs, is what the company uses to determine your haplogroup and the group you belong in for your projects and things like that. The short tandem repeats are when a set of 3 bases repeat over and over and over. And you can see in the first example for man number 1, the STR, the CTA, repeats over and over and over 5 times. In the second man, it repeats 6 times. In the third man, it repeats 7 times. So the STR testing is different than SNPs. If you look on the left, the SNP is where one base is substituted out for another base. So we have A's, T's, C's, and G's, and they form sequences. Those are bases. They pair up, they form sequences, and the sequences is what the DNA testing companies read. And so the SNP is when a base is in there that's different than the majority of people who have that location. So most men, in this case, where the example it's showing, most men would have an A at that particular location, and the SNP test is determined that there's a T there instead. So there are a lot of reasons why, SNPs happen, mutations, bases substitute in. There's a lot of things that happen when DNA is copied and copied and copied. It doesn't really matter if there are 5 or 6 or 7 repeats. It just matters that it did happen. So it doesn't matter what those bases are or the fact that there are 5 of them or 6 of them or 7 of them. What matters is that the value is different than your closest match. And what matters is how is it different. So man number 1 and man number 2, well, that's not a good example. Let me use man number 2 and man number 3 that have the same SNP. Man number 2 has a repeat of 6. Man number 3 has a repeat of 7. So they're one genetic distance from one another. So when my husband did his initial testing, the Y37, we received an email, and the email said, you may be able to find further results, further out on the tree, so to speak, if you buy more SNP testing. And the SNP pack that we purchased for the M253 group was $99. And I love how they say, get 174 SNPs related to this group for only $99. So yes, we spent another $99 to get him further out on the tree. So you will receive an email, and if someone you know is tested and maybe they tested a while ago, you can inquire with the company if they have any SNP testing available that would push you further out on the tree. I'm sure they would be glad to sell it to you if they do. So how do you use Y-DNA results? There are a couple of ways that you can use them individually and use them in aggregate with others. You can join a Y-DNA surname group or a haplo project or a regional project. There are a lot of those on FTDNA's site. There is a tab at the top called projects where you can find one. Also, you can work on your genealogical tree, your paternal line. So you can use those matches and some of their information and their surnames as clues for you. So let's say that you test and you are a Bergheimer and you join a group and there aren't any other Bergheimers in there. You might want to start investigating some of the other surnames that are in there, because perhaps there's a misattributed parentage or some other event that has you linked to those particular men. Look at the locations of matches. I think one of the fantastic things about FTDNA's website, SNP Tracker, and some of those others is the migration patterns, the information that we can glean from the maps of our matches, from the locations of our matches, and that is all very important information. And just keep in mind, if someone has the exact same Y DNA as you or your husband or whoever tested, they are in your father's line. They are in that line. So it's really fascinating to be connected to men around the world who are all connected genetically through that one particular Y DNA segment. Y-Search is another place where you can use your results, and you can search by last name, you can search by matches and haplogroups. There are tools there that you can use. I tried to search for Bergheimer, and you can see here there's no records found, no pedigree, but I could upload some things here and make my husband available to be searched, and maybe there would eventually be something that results from that. I do check it every once in a while just to see if someone's in there. But I know in the US, my husband has only one first cousin, two nephews, and one son. So it's a small subset of men. And if they've tested, I probably would have sent them the test. You can learn more through surname groups, ysearch.org. You can join a geographic project at FTDNA, a haplogroup project, a surname project at FTDNA. The group volunteer administrators are amazing, and they will help you. So if you don't understand your results, if you're not sure what it means, one of the first things they'll have you do is change the share settings so that they have access to your DNA matches, or I'm sorry, to your SNP testing and your STR testing, and they can add you into the project using that. There's also a book that have the cover here by David Vance. It's a pretty recent book, came out in the winter of 2020, The Genealogist's Guide to Y DNA Testing for Genetic Genealogy. It's a really fantastic book, particularly if you're a visual learner and you want to learn about the charts and the history and what do you need to know from 0 to 10. It's really a fantastic book. I enjoyed that a lot. Also, isog.org always has is kind of a go-to information place that's free. ISOG's wiki page, they have isog.org/tree that has some great information. So nose around at ISOG, you'll be able to find some things. DNA Central is Blaine Bettinger's site. That is a paid site. Also, there are some other free resources, always YouTube videos, things like that on Y-DNA testing. I've heard a lot of really great Y-DNA webinars from Diane Southard. So you can find resources, just keep looking. And if you're not sure, you can email me and I'll send you some suggestions. So understanding your matches is all about mutations. What causes those mutations? DNA makes copies of itself, and sometimes the copy isn't quite a perfect copy. There are some environmental factors that cause mutations. I'm sure that there are causes of mutations that we just haven't scientifically uncovered yet, but we do know that UV light, chemical carcinogens, X-rays, radioactive substances, things like that can cause mutations and often do. Also, there are other kinds of mutations that can cause substitutions, insertions, deletions, and inversions. That's a little bit more biologically intense than what we plan to talk about today. But I do talk about DNA health reports where substitutions, insertions, deletions, and inversions often will cause health problem in someone, if they have too much DNA or an extra copy of something, or something's been substituted instead of the typical DNA or the typical gene at that location. So you'll hear more about that if you watch any of my presentations on DNA health. Remember, we're always talking about deep ancestry versus recent ancestry. The timeline is important. The mutations can happen at any time. They can be reversed at any time. Genetic distance of 1 can be hundreds of years old, or it can be 1 generation. So 2 brothers can be a genetic distance of 1. A father-son can be a genetic distance of 1. But the most common case is a genetic distance of 0 between your brothers and your father. And over generation, the number of repeats in the STRs can change up or down, and the changes create individual lines of men. And you see that most often on the large reports and the project reports. The haplogroup shows your deep ancestry and relative relationship to the first man, and haplogroups with STRs show ancestors in more recent time. So there are different ways to view Y-DNA, and you always have to kind of keep in the back of your mind whether or not it's genealogically relevant in recent time. I'm going to show you this again because I think it's really important to point out that there are 2 different kinds of things that are measured in the Y-DNA testing. STR, short tandem repeats, so that's where sets of bases repeat. SNPs are single nucleotide polymorphisms, and what that means is that one base subs out for another base. So I just want to point out, and that those are true. The SNPs are measured in autosomal DNA as well. So just want to point that out. On your report or on your certificate, FTDNA calls them certificates. They have a series of codes, and sometimes it's confusing, like what do these codes mean? But the chromosome segments all are numbered, and then they have STR values. So DYS is a chromosome segment of the Y-DNA. 393 is the marker, so that's the specific location. And it has a value of 15. That means that that series of bases repeated 15 times. So the coding has to do with the segment and the marker and how many times it repeated. So this is what the certificates look like. I just have a blow up here of the STR certificate. So this starts with DYS 393, and my husband's value is 13 repeats. And then there's a DYS 390, and his DYS 390 has 22 repeats. And then you might notice that there is further across DYS 385 that has 13 and 15. It doesn't mean 13 to 15, which is the way that I thought it was when I first looked at it. 13 is one of the values and 15 is the other value. So what happens in your Y testing when it's read from end to end in the same direction every time, and one of the DYS 385 locations has 13 repeats, and there happens to be another DYS 385 that has 15 repeats. And there are locations where there is multiple of those that are checked. So what does 37 markers mean? So I told you my husband took the 37 marker test. So this is the first 12 that are checked. And I think you can see there, that's the first row of the certificate. This is the second set that are checked. And you can see that that's the second line of the certificate. And to get up to 37 markers, there is a third line that goes from DYS 460 to DYS 438, sorry. So the 12 test is the first line, and the 25 test is the second line, and the 37 test is the third line. Can you see that on the white on the right? So the 1 through 12 is the 12 test. The 25 tests includes 1 through 12 and 13 to 25. And then the 37 tests includes the 2 prior and then the additional 37. So every time you upgrade your test, you get more and more STRs. So the 67 test would include all of these here and the additional. So why are there 2 values at that DYS 385? That's because it's a multi-copy STR. So there are multiple versions of that STR in different locations. And each location can have a different number of repeats. The STRs on this list correspond to the STRs in the Y-DNA projects. So this is just a small look at the STR project that my husband is in. But you notice that the DYS 393 is the first one in the first row of the certificate, and it's the first column that shows up on screen here. And the second marker is the second column, and the third marker is the third column. This is something that really helped me understand Y-DNA testing and those huge charts that are generated by the projects. And I think I mentioned when this first started, my husband did this Y37 test. He was kind of a party of one for a really long time. There really wasn't a project to place him in. And then he ultimately was able to be placed into a project. So now I have a project sheet to look at that I've never had before. So for me, the information on the certificate was rather meaningless until I was able to correlate that each item in the certificate corresponded to a column on the chart and his values versus other men's values on the chart. We do need to talk about the one-step mutation again and two-step. I just want to point that out because we did talk about it earlier on, but this is what it looks like in the chart. So you can see here that there are 2 men where they have a genetic distance or a one-step mutation between them. So all the other men in that column have 11 repeats, and then 1 man actually has 12 repeats at that location. So that's the kind of information that's captured by the project, are those subtle differences between the men in the project. You can see here that this column has mostly 28s, but not always, but the difference between 28 and 30 is 2. So that's a 2-step mutation between those 2 men. And so the project administrators are placing men on this chart according to how much they vary with the other men in their group. This is the SNP certificate for my husband, and the SNP certificates are going to look very different depending on which branch of the tree you're in. The the short tandem repeat certificates are going to look the same based on whether you did a 12 test, 25, 37, 67, and so on. But the SNP certificates are going to look very different. So this is the list of the SNPs that were tested. He was placed into this group. You notice they do reference the IM253 lineage, which is the original group he was placed in. And it'll tell you whether you're positive or negative for certain SNPs. So whether you do or don't match the group, and then how far out you are on the branch. And I just want to show you kind of another visual of what those mutations look like. When bases are replaced with other bases, that's a SNP. And again, that happens in autosomal DNA too. So this is the Y-DNA project for the parent group IZ140, which is a few branches back from where my husband's terminal SNP is. And so eventually I've given them permission, the administrator's permission, to add his data in here. So eventually I'll be able to see him listed in here along with all the other men who are on that same haplogroup and the same chain. And you notice that right about a little bit to the right of center of the screen, they always have the STR markers in the order they appear in your certificate. So the DYS 393 is always going to be the first one. So you'll see those who've done the 12 test and then some data drops off. You'll see some who did the 25, some data will drop off, and then you'll see the 37, 67, 111. So how do you use all this? Surname projects, haplo projects, look for matches and investigate all of them. Look at your matches trees, see if they have family finder results. If you only did the Y test, you might want to also do an upgrade and do the family finder results for yourself. Your Y DNA match might not match your autosomal DNA match for your cousins in recent time, and that's not unusual. So just keep that in mind. You should have autosomal matches again, always to first and second cousins, but not necessarily to third cousins and beyond. You might want to try targeting testing, and you might want to wait for more Y DNA matches because you just never know. So again, my name's Kelly, and if you have any questions, you can contact me through email, and thanks for joining in today.